Let's talk about the fidelity of DNA replication. So fidelity is another way of saying the accuracy, the faithfulness or the accuracy of DNA replication. It's very important that our genomes be replicated very accurately. Otherwise, you get mutations, and mutations, most mutations are neutral. A few mutations are beneficial, but a lot more mutations are harmful than are beneficial. So we want to keep the mutation rate down, and accordingly, DNA replication is extremely accurate. Usually when you replicate a genome, you only make one mistake every billion base pairs replicated, right? So in the E. coli genome, where there's only 4 million base pairs, then you're only going to make one mistake in every several hundred cells, okay? In our cells, when we replicate the genome in one of our cells, we're going to make 3 or 4 mistakes, okay? But that's an acceptable error rate. Okay, but so how do we achieve this high level of fidelity? As we've seen, the bases are selected during DNA replication by virtue of a Watson-Crick interaction. But it turns out that some non-Watson-Crick base pairs, such as this GT base pair, which we've looked at before, are only about 100-fold less stable than a normal Watson-Crick base pair. So this base pair is definitely less stable than a normal Watson-Crick base pair. We've seen that's because it doesn't have correct Watson-Crick geometry. The pyrimidine residue is raised up relative to the purine residue, and so in order for the duplex to accommodate this GT base pair, that results in a distortion in the helical backbone, and that's energetically unfavorable. Okay, and so as a result, this base pair is 100-fold less stable than a normal Watson-Crick base pair. So if that's all we had to rely on to ensure fidelity of DNA replication, then we would expect an error rate of 1 in 100 instead of 1 in a billion, and life would not be possible as we know it. Okay, so how do we explain the 1 in a billion error rate as opposed to the predicted 1 in 100 error rate? Well, it turns out that this low error frequency is accounted for by multiple redundant safeguards. Okay, and so there are 3 of them that I'm going to be talking about. So it turns out that the binding pocket of DNA polymerase clamps tightly around the base before catalysis, and so wobble pairs, so we sometimes would call this a wobble pair, they simply do not fit into the active site. Okay. DNA polymerases also have ways of recognizing when they've made a mistake and then trying again. So DNA polymerases have editing exonuclease activity that allow them to erase their mistakes and try again. Okay. And finally, cells contain mismatch repair systems that come along after DNA polymerase is done to clean up any remaining errors. Okay. Okay, so each of these safeguards then improves the accuracy by about 2. None of them is perfect, but each of them improves the accuracy by about 2 to 3 orders of magnitude, and that's how you get from a 10 to the minus second error frequency to a 10 to the minus 9th error frequency. Okay, so let's talk about these 3 things one at a time. So here is another view of Clentac again, the Thermus aquaticus equivalent of the Klenau fragment. Okay, and what we see is the Clentac in 2 different conformations. It turns out that during the catalytic process, with the addition of each nucleotide, Clentac cycles between a so-called open conformation, which we see here, and a closed conformation, which we see here. So in the open conformation, then there's a big space between the thumb and the fingers. Okay. In the closed conformation, the thumb and the fingers come together to clamp over the substrates. Okay. And so we need to open up the active site. We need to be in the open conformation for DNDPs to be able to diffuse in there and bind in the active site. So that's why we need to open up the active site. We need to access the open conformation at each catalytic cycle. Okay. But then before catalysis can occur, before that chemical reaction can actually occur, we need to close the active site. Because it's the process of closing that active site that pushes the nucleotide down into the proper position in the active site so that catalysis can occur. Therefore, catalysis only occurs after closure. Okay, and so when we close the active site, we push that nucleotide down into the proper space for catalysis. In the process, we also constrain that nucleotide in a very tight space so that only the Watson-Crick base pairs can fit. Non-Watson-Crick base pairs don't have the correct fit, the correct shape to fit into the active site after closure occurs. So usually, most of the time then, catalysis cannot occur in the presence of mismatches near the growing end of the duplex.